And again,
क्या है यार, ये क्या चीज? ماذا؟ لو بكنيني كسرنا يا في سابدي.
ميم تو هذا تيلر كون
إن أنا هذا ما خلي جونيور دي.
Elaka Berula Mina. Berula? That one design thing, you know.
Ah wow.
ده څه شی دی لکه
دا خو لوی مخلوق دی باچا.
Elaka
عمر وځي ځکه
Idrisa.
ها
وایا
کمرانه دا څه دی لکه
گورنر وایا
الکه ومنه پیجن دی باچا. لکه دا څه شی دی؟
ये क्या चीज है यार, ये आप लोगों ने हमें इनवाइट किया, हम तुम बताओ हम डिस्टर्ब हो रहे थे.
Idrisa. Idrisa.
जी
hello, dear guys.
जी
So
वालेकुम सलाम.
here we'll start our conference. So one minute we'll connect all who want to take part in it. So just wait a little bit, and then we'll start.
Okay, sir.
So all the participants should be here. Najula here.
Yes, sir.
I can see you. Ibrahim Khan? Not here. Zainul Abidin? Muhammad Zainul. I see you here. Then Naim Nimra.
Yes, she's just training.
Rabia.
Yes, sir.
Yes, they are waiting. Okay, we'll continue later. 2 minutes.
Ibrahim de taso golli mana. प्लीज यार कंजल पर किम्मा को इदारे काट की
Yes, sir.
Probleme?
जी जी
اسوي صار اسوي
सर वी वेट ओके लाइक
Oh, I see organizers of conference are already here. So most of students also joined who take part in this conference. Okay, here I'll be like organizer of this conference. So Salымбеков University provided the conference in oncology. So the full name of this conference is named The Modern View of Pathology of Oncological Diseases. And I wanna greet you here for this interesting problem. And for first word, I wanna give a word for the respected sir. He's Doctor of Medicine. He's a prorector of Educational Department. It's Tuлекеев Т.М. He is Vice Rector.
सर प्लीज allow the students.
Yeah, during this conference I'll allow it. So and first word going to prorector, please.
Спасибо. Уважаемые участники студенческой конференции, посвященной актуальной проблеме биологии и медицины.
Respected participants of Student Scientific Conference, this conference is enlightened for a very actual biomedical problem.
Современные представления о патологической физиологии и морфологии онкологических заболеваний.
It's a modern view of pathology, pathological physiology, pathological morphology, and histology of malignant tumors.
Известно, что онкозаболевания наряду с сердечно-сосудистыми заболеваниями занимает ведущее место в реестре Всемирной организации здравоохранения.
It's well known that tumors, along with cardiovascular problems, cardiovascular diseases, they are one of the main leading position in the World Health Organization list of mortal diseases.
На долю страдающих онкозаболеваниями приходят контингенты разных возрастов, в том числе молодого поколения.
So patients with oncology diseases are of different age and even young age too.
Пато, морфо, гисто и иммуногенез опухолевых процессов тканевых структур вариабелен соединительной, нервной, костной, мышечной ткани, либо крови.
So oncological process pathologically, morphologically, histologically, and immunologically vary, including epithelial, connective tissues, nervous tissues, blood tissues, etc. So it very vary.
Между тем, для студентов-медиков или для будущих врачей имеют глубокий смысл осознавать важности современного представления.
It has a very deep meaning to recognize importance of modern approach to study tumor process for medical students and future doctors.
Об этиопатогенезе, перспективы диагностики и лечения онкопатологии, и трансдифференциации тканей. изменениях микрофлоры, морфологии нейронального сигнала, иммунных реакциях организма, а также вопросы нанотехнологий новообразования, включая генной терапии, фармакогенетики и другие.
It includes etiology and pathogenesis, perspectives of diagnostics and treatment of oncopathological process, diseases, dedifferentiation and transdifferentiation of tissues, alteration of neuronal signaling, immune reactions against tumors, nanotechnological approach in understanding and treatment of tumor, gene therapy, pharmacogenetics, and other having a theoretical and practical meaning.
Уважаемые студенты и участники конференции, мы понимаем, что программа конференции не дает полного ответа на медико-биологических, социально-экономических и других аспектах онкозаболеваний.
Dear participants, we understand that this conference does not give an answer to all medical biological questions appeared in oncology service, including socio-economical and other points of this complicated problem as malignant tumor.
Однако можно полагать, что от полученных информаций вы приобретете определенные теоретические знания и направления об этом недуге.
But we can think that this conference will deliver you a huge bunch of information in this very challenging field of knowledge.
Ректорат, деканат Международного факультета медицины Учреждения Салымбеков Университет желает успешного проведения студенческой научной конференции.
So administration of dean and vice rectors and rectorate of international medical faculty of Салымбеков University wish you success in leading of this conference and gain a new knowledge.
Уважаемые участники конференции, студенческую научную конференцию объявляю открытым.
So, dear students, let's make this conference open. Okay, thank you, Dr. Zamildalovich. So we open our conference. So now I want to give say a few words to organizers of this conference, organizers of this reports. It's Ismail Bermet, Sagimbaeva Bermet, and Оморова А.С. So please.
Hello, dear participants of today's conference, dear students. It is a great honor for us to present to you at today's conference new approaches in cancer treatment. First of all, we would like to quote Avicen. Medicine is to preserve health and cure disease arising from causes affecting the body and producing symptoms. So now, at today's conference, we will consider and discuss overall information about cancer, cancer types, hallmarks, known hallmarks, as well as new ones, which was revealed in 2021. And we will dive into new approaches and techniques in cancer treatment. And I want to give the saying of own speech for Med.
Thank you, Ms. Aydana. Hello to everyone and all organizer of the whole conference. I'm glad to see you and all of the participants as a face of the students. I'm also glad to see you. And today conference, I'm just try to repeat it again. This is related to the cancer. What is it cancer about? What are the types? And in today's conference, we try to figure out what are the new treatments and approaches. And our conference is conditionally divided for two parts, which first will be for the introduction, and the second part and the types of the hallmarks and new hallmarks as well. And the second part will be about the known treatments that have been used until now and new cutting edge techniques and the breakthroughs in the cancer treatments. So now we are glad to introduce you to our dear students who will be shed some light in understanding of our meeting today. So first we'll be starting by Najibullah. Najibullah, are you here?
Yes, ma'am.
So Sorry for interruption. One wish for students. So please, we have reglement. Yeah, not more than 7 minutes. One report, not more than 7 minutes. So I understand your report is very important and large, but please not take it only 7 minutes, not more, please. Yeah.
1 second, I will just.
We need some time to open the presentation. 1 minute, please.
Регистрация экрана. Видно? Okay, Najiullah, please start. Hello, Najiullah.
Hi and assalamu alaikum everyone. So today my topic is cancer, its introduction and pathophysiology and types. So first of all, I will tell you that how common is cancer. So according to National Cancer Control Program, so there are nearly 2.5 million program. There are nearly 2.5 million cancer patients in India, and about 0.7 million new cases are diagnosed each year, and nearly half of them they are die. So more than 60% of these patients are in the age of 35 to 60. So when the prime of their life, it is expected that with improved expectancy, the number of cases will triple in the coming years. So about one in 4 of us will get cancer, and about 1 in 5 of will die due to cancer. So first of all, we have to understand cancer. What is cancer? And so, like the difference between the normal cancer and the normal cell in the cancer. So the normal cells, there are trillions of cells in our body, and the healthy cells, they are programmed because they know that what to do and what not to do. So then, there's some pictures about that which show us about the cancers, like how it grows. Forward it, ma'am. So then the normal cell and cancer cell, there's a difference between normal cell and cancer cell in a mono layer. Yes, ma'am. So look at this is the normal cell which grow in a mono layer, and the down one is the cancer cell which grows like a clump in a clump. Go forward, ma'am. This is also one of the picture of the normal cell and the cancer cell. So then this is the division of the normal cell and the cancer cell. Like the normal cell, they divide like like in a proper way, like in a program way. And cancer cell, it divide like we can't imagine how much it goes forward. So then what is cancer? So cancer is a general name for more than 100 different diseases. The common point about all these different diseases is that a particular cell of the body is growing out of control. And the special thing in the cancer is they do not know what to do and when to do. So the division use of all the resources. So some commonly used term is we use for the cancer is neoplasm, which we give the new growth, the tumor which is swelling caused by neoplasm, and the benign neoplasm, and which is spread in like a particular body, and a malignant is spread in the whole body. Then we have type of cancers. So we have different type of cancers in the whole body. Like we have cancers in the lungs, we have cancers in the breast, we have cancers of bloodstream, of lymph nodes. Also, we have fat, bones, and the muscles. Like in the Tobago causes 14 types of cancers. This is the cancer of nose, in mouth, larynx, esophagus, lungs, liver, pancreas, stomach, bladder, ovary, and leukemia. So the National Cancer Institute gives the common types, like we have bladder, lung, breast, endometrial, leukemia, pancreatic, thyroid, prostate, colon, rectal, lymphoma, kidney cancers. So this is the example of primary cancer, and where they can metastasis spread to, like the skin melanoma, then it goes to the brainstem. The bone metastasis, it also cause the brainstem, the lung cancer, the liver metastasis, and the pancreas cancer. Then we have most common cancers in men, women, and children. So in the men, we have common cancers are prostate and the lung and the colorectal. In the women, we have breast, lung, and colorectal. In the children, we have leukemia, brain tumors, and lymphoma. Then we have a carcinoma, which is begins in the in skin that line the internal organs like skin, lung, colon, and pancreatic. And their subtypes are endocarcinoma, basal cell carcinomas, squamous, and transitional cell carcinomas. Then we have a sarcoma, which begins in the bone, cartilage, and other connective tissues. And there are two types: the bone sarcomas and the soft tissue sarcomas. Then we have leukemia. The leukemia begins in the blood-forming tissue. They cause large number of abnormal blood cells and enters the blood. It is around the age of 0 to from the 5 to 5, 14 to 18, and 19 to 40, and about 60 plus. Then lymphoma and myeloma that starts from white blood cells. They affect lymph nodes of specific sites like stomach, brain, and intestine, and there's also of two types: the B cell and the T cell lymphocytes. So myeloma begins in plasma cells type of blood cancer, and their plasma cells are capable of producing various antibody. It can damage the bone, kidney, and red blood cell count. Then we have central nervous system cancers. They start with the brain and brain tissue and spinal cord. The brain controls the body by sending electrical message along nerve fibers, and brain and spinal cord from the central nervous system. Then the pathophysiology of the cancer. So the cancer is ultimately a disease of gene in order for cell to start dividing uncontrollable. Genes which regulate cell growth must be damaged. The proto-oncogenes are genes which promote cell growth and mitosis, and tumor suppressor genes. So discourage cell growth or temporarily halt cells division to carry out DNA repair. But typically a series of several mutations to those genes are required before a normal cell transforms into a cancer cell. When normal cells are damaged beyond repair, they are eliminated by apoptosis, and cancer cells avoid apoptosis and continue to multiply in an unregulated manner. So difference in cell growth. So most normal cells are limited to approximately 50 to 60 generations or division before they die. So the programmed death is called synthesis, and its mechanism is controlled by the cell's normal biological clock. So this biological clock is called telomere. So the function of the telomere is to protect the chromosomal ends of the DNA from damage. With each replication, the telomere is copied completely. With the age, the telomere grows copied completely. With the age, the telomere grows progressively shorter when it shrinks below a certain level. Next, ma'am. So carcinogenesis. So it's the process during which normal genes are damaged so that the cell lose normal control mechanism of growth and proliferate out of control. When the genes of a single cell are altered by a carcinogenic event, the offspring of the single cell continue to mutate and divide, produce even more virulent mutant clones. There are three phases of carcinogenesis. We have initiation, is the conversion of normal cell to tumor cell. Then we have a promotion, is the process of altering the genetic expansion of a cell by increasing DNA synthesis, and also increasing the number of copies of a particular genes and nurturing the intracellular. Then we have progression, is an irreversible phase in which the cells are led to morphological changes and malignant behavior. Next one. So this is the process of carcinogenesis. Like we have divided the phases into three. So then there is a difference between the benign growth and the malignant tumor. So the benign growths, they are encapsulated, non-invasive, limited growth, and doesn't metastasis and rarely lethal. The malignant tumor that are not encapsulated, invasive, uncontrolled growth, and metastasis and often lethal. So what causes cancer? So we have a lifestyle activities, the environmental, and a family history. So the main causes of cancer is the environmental factor, which are the tobacco, alcohol, dietary factors, viruses and parasites, and we have genetically leukemia in monogamous. So the lifestyle risk, the lifestyle the lifestyle we have the main causes are smoking, with a diet high fat and low in fruits and vegetables. So lack of exercise, obesity, unprotected exposure to the sun, ultraviolet rays. environmental risks. So second, hard smoke industry, air pollution and industrial pollution, and also chemical exposures. Then we have inherited risk. Less than 15% of cancers are inherited. Gene mutations are linked to some inherited cancers. So the cancers that may be caused by inherited genes mutations are colon cancer, breast cancer, ovarian, prostate cancer, and skin cancer. Then we have preventive measure of cancer. So we have to do physical activities, avoid obesities, healthy dietary practices, and reducing occupational and environmental exposures, reducing alcohol uses, immunization against hepatitis B virus, and safe sexual practices for avoiding cancer genesis. Fruit and vegetable decrease the cancer risks. So cancer rates could decline by up to 20% if everyone consumed 5 fruits and vegetables a day. So cancer fighting substances are antioxidants, dietary fiber, carotenoids, and flavonoids. So limit alcohol to not more than. So avoid alcohol drinking, the use of unnecessary drugs, protect relationship, disease is not a friend. Avoiding smoking or chewing tobacco. So cigarette smoking is the leading cause of preventable death. So what is staging of cancer? So staging means assessment of the extent of spread of cancer inside and beyond to its site of origin. So we have local disease is cancer limited to its primary site, regional disease is cancer spreading to regional lymph nodes, and distant disease is cancer spreading to the systemic organs like bone, brain, and lung. So staging of cancer, we have divided the staging cancer into 4: the mobile primary and secondary, the fixed primary, and the distant metastasis. So why do we stage cancer? So to choose the best method of treatment, and late cancers are too advanced to be resected. So chemotherapy and radiotherapy may be used to assess the prognosis. So the method of staging of cancer. So clinical methods, radiological methods, pathological methods. We do mammography, X-rays, ultrasound scans, CT and MRI scans, and a position imaging tomography. So the clinical method examination of the cancer bearing sort for. So size, T, large diameter of cancer, mobile or mobility, mobile or immobile cancers, skin overlying cancer, intact endometastasis, and ulcerated. So examination of draining lymph nodes. Pathological methods, the final diagnosis of cancer is based upon the histopathological examination. The final need respiration strategy or through histopathological examinations. Then we have radiological methods. We have do mammography, plain X-rays, ultrasound scans, and CT MRI scan and isotopic scanning.
I'm sorry. 1 second.
Jalal.
Who is Jalal? Игорь Алексеевич, мы извиняемся. Что это такое?
Да-да-да, сейчас я удалю Джалала отсюда.
Ага.
Guys, a little bit more respect for the students who are doing presentation. What is a childish attitude?
Sir, I am there. Anyone who joined from my name, sir, he is abusing.
No, this is not yours.
Jalal.
Yes, ma'am. I am Jalal.
Oh, okay, it's fine.
Anyone has joined from my name.
So this is the worsening situation. Mm-hmm. Sorry for interrupting, guys. Give us a couple of minutes. Mm-hmm. Shall we continue? Nigella, are you ready?
Yes, ma'am. Exactly.
Yes, please, my friends, continue.
1 second. Игорь Алексеевич, вам видно презентацию?
Mammography, ma'am. No, we can't see the presentation.
Почему-то я ставлю, а у меня скидывается. Вот сейчас нормально. And Najullah, may you...
Okay, mammography. Mammography is used a low-dose X-ray system to examine the breasts. So mammography is used as a screening or a diagnostic tool to detect early breast cancer. The down one can detect abnormal areas of density, mass, or calcification. They may indicate the presence of cancer. Then screening mammogram. So mammography can show change breast up to 2 years and before a patient or physician can feel them. So screening mammography is done annually begin at age 40. So women who have had breast cancer and those who are at increased risk due to a genetic history should begin screening before age 40. So diagnostic mammogram. Diagnostic mammography is used to evaluate a patient with abnormal clinical findings such as a breast lump that have been found by the woman or her doctor. So diagnostic mammography may also be done after an abnormal screening mammography in order to determine the cause of the area of concern on the skin exam. So plain X-ray. A chest X-ray is often used to determine whether the cancer has spread to the lungs or not. Then we have bone survey, scaly metastasis, which is also done for the examinations. Then we have used ultrasound of the breast, like we have the exam of the cancer and the cyst. Then we use CT scans, the chest CT scan. This image shows a 5 millimeter tumor in the right cardiopharynx lymph node, and the image shows a 1 centimeter tumor in the right retrocoronary lymph node. Then CT scans, abdominal CT scan, liver metastasis and RN metastasis. Then MRI scans, we also use the MRI scans for the cancers. Then PET scans, the PET is the position imaging tomography. So they are still in the experimental phase and are one of the newest breast cancer diagnostic technique. So a small amount of radioactive material is injected, and which often indicate rapid cancer growth. Take up the radioactive material. So normal PET scan, which shows the normal heart, the normal left kidney, and the normal bladder, and also shows the normal aortic kidney. The PET scan showing abnormal lymph node, then so we can see it's all the rough, and we can't we really show that this is the cancer and this is not. Then PET scan showing in women with breast cancer that has spread to bone. So this is the image of the breast cancer which is spread to the bones of the woman. So conclusion, we get the screen mammography can detect precancerous changes in the breast, and staging of cancer requires clinic and radiological investigations. The histopathological examination of tumor tissue is the only confirmation of malignancy. Then hallmark of cancer. So we have 8 hallmark of cancer: the sustaining proliferative signaling, evading growth suppressors, resisting cell death, enabling replicative immortality, inducing angiogenesis, activating infusion and metastasis, and deregulating cellular energetics and evading immune destructions.
Okay, thank you very much, Najiullah.
You're welcome.
Students who are participating in the conference, do you mind if you open your slides from your side, because we don't know whether you want to stop or maybe you can show something. All right, are you okay with that?
Yes, okay, ma'am.
All right, so next student will be representing us the hallmarks of cancer will be Ibrahim Khan.
Yes,
so your turn, please start.
Slide. I will share screen, ma'am.
Yes, please. Они могут, да?
Ma'am, please.
У них есть доступ, возможность? Так будет лучше.
Are you see my
no,
screen, ma'am?
Not yet. Ibrahim, please try to explain short, but...
Are you see my screen, ma'am?
Not yet. Every student try to fit in 7 minutes. Cut it down if it is necessary. You have to highlight the main points. Otherwise, we will sit until tomorrow. Yes.
Yes, ma'am.
Yes. 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 Good.
Are you see, ma'am, now? Hello, ma'am. Okay, ma'am. Today my topic is new hallmark, dedifferentiation and transdifferentiation, ma'am. In in 1957, Konrad Weidenker proposed the unidirectional development model, where in pluripotent stem cell at the top of the hill progressively lose their pluripotency as they follow development pathways and end up among differentiated values in a terminally differentiated state. However, the concept of the tumor cell plasticity goes against the Weddington landscape, where de-differentiation allows non-cancer stem cells to
Ibrahim,
acquire.
Ibrahim, sorry for
stem cell
interrupting. Can you speak louder,
replacement.
please? A little louder, sound up.
Yes, ma'am.
Thank you.
And sorry, one
Yes,
more
ma'am.
request. Please
Now, sir.
arrange your phone horizontally, please. Arrange your phone horizontally.
What you say, ma'am?
Arrange your phone horizontally, so because most of us, it's recording to website, so that's why we need to see.
The screen दिताओ का wrong का screen.
Rotate your screen. Rotate your screen.
Okay, ma'am. Now.
That's
Now,
better.
sir. Is it okay, sir?
Yes, it's the best.
Okay, ma'am. In 1962, Sir John Gordon challenged the unidirectional dogma of development with his ground-breaking study, which showed the formation of a fully functional tadpole clone, even when the nucleus of a prozygote was replaced by a nucleus by a harvested from a terminally differentiated tadpole intestinal cell. This proved his hypothesis that the genome of the mature specialized cell has all the information required to develop into the different cell types of an organism. However, a Gordon experiment involved physical removal and transfer of the cell nuclei, as such as a question remained whether such a hypothesis could be replicated in intake cell. 40 years later, this question was answered with a proof of concept study, which defined as a Weddington landscape in intake cell, inducting just poor genes. The Yamanaka factors, C-MyC, Cropel-like factor, 4KLF, sex determining region, Y box 2, SX2, and octomer binding transcription factor by third 4. Tagashi and Yamanaka were able to develop what they term induced pluripotent stem cell, which had the ability to differentiate into any of the cell lineage names, lineage or endodermal, ectodermal, or and mesodermal. This formed the basis of the hypothesis that tumor cells, which are champion survivors, will hack and mechanism in order to survive as such de-differentiation is a electorate hallmark for them to achieve immortality. Cancer stem cells are a unique subpopulation which possess
Ibrahim? Net problem.
हालांकि कलिया वाला आवाज न राजी
Игорь Алексеевич, а как быть? Продолжаем следующего или подождем?
Давайте, наверное, подождем.
Мгм.
Нет, давайте следующее, потом Ибрахим продолжит. So let's continue. Ibrahim will continue later because of technical issues. So Zainul Abidin here.
Yes, sir.
So Zainul, I hope you're ready. I hope you have no important issues. So start your presentation. So let's introduce Muhammad Zainul Abidin, GM 1119, topic epigenetic dysregulation. So again, Jalal, yeah?
Здрасте. Come on, this is not funny.
तेरा कदा से भी
वही
जैनुल आबिदीन, ये क्या हो रहा है?
Jalal, I hope you feel yourself very happy. Zainul Abidin,
Yes, ma'am.
you can start.
Ma'am, can you kindly share my presentation? I am having trouble.
Okay, 1 second. Абидин скидывал линку тебе? Абидин тебе скидывал? Мне нет. Заин. Да, вот он. Скинь мне. I'm sorry, 1 second. Выведи. Заин напиши, что вы.
ब्रदर जी जो भी है ना, ये ठीक काम नहीं है, टीचर्स की इंसल्ट है, बराए मेहरबान ऐसा ना करें।
Start, please.
Yes, ma'am. Actually, epigenetic dysregulation.
I'm sorry, I'm sorry, I'm sorry.
No, ma'am, it's okay. The differentiation of progenitor state is the rate limiting step in the melanoma formation, but it is the underpinned by
I'm sorry, I'm sorry.
No, ma'am, it's okay. Yeah, but it is underpinned by epigenetic machinery, although Yamanaki factors provide the possibility of reprogramming differentiation somatic cells to the pluripotent state. The blockage of histone H3 lysine 9 methylation has been shown to enhance this reprogramming capability. Similarly, in the context of DNA methylation, another key epigenetic alteration, the promotion of DNA demethylations via stimulation of 1011 translocation enzymes using vitamin C enhances reprogramming to a pluripotent state. Epigenetics can also regulate the process at binding back to clock to a pluripotent state on the basis of chromatin state and the expression level of chromatin modifying enzymes, providing a conceptual link with our first hallmark of differentiation. Yes, ma'am. What is epigenetic? The term epigenetics was first coined by Conrad Weddington, which is defined as a branch of biology which studies the causal interactions between genes and their products, which bring the phenotype into being. And after then, the Holder refined the definition of epigenetics as the heritable changes in the gene expression without altering in the DNA sequence. That is, altering the phenotype without altering the genotype. Yes, ma'am. Next. Yeah. Epigenetic fingerprint. As we discussed the features of hallmark epigenetic dysregulation in an active functional capability, it is a unique feature among cancer cells, and there are epigenetic fingerprints on tumor cells which reflects its chronic nature. Epigenetic dysregulation viewed as a bystander would be a relegation of its active role in tumorigenesis, as several studies have pointed out its role in tumor initiations. In 2006, Feinberg proposed the epigenetic progenitor model of tumorigenesis, wherein epigeneticization of the progenitor cell population give rise to tumors. Next. Yeah, sustained proliferative signaling. It's actually a high complementary hallmark capability for sustaining proliferative signaling in cancer cells.
One moment, please.
Yeah. In cancer cells is the ability to evade growth suppression. Several tumor suppressing protein coding genes that operate in diverse ways to inhibit cellular growth and proliferation has been discovered. Just 1 minute. Come on, back. Yeah. Evading growth suppressors. It's actually a high complementary hallmark ability for sustaining proliferating signaling in cancer cells is the ability to evade growth suppressions. Alternatively, tumor suppressors genes could also bind to other protein expressed in tumor cells and therefore become inactivated or rapidly degraded. Invasion and metastasis. An integral component of hallmark of invasions and metastasis is an reversible epithelial mesenchymal transmission, or abbreviated as EMT, orchestrated by the interaction between epigenetic modulators of chromatin configuration and EMT, including transaction factors, transcription factors, expression of ehedrine. A key coordination of epithelial phenotype is the lost during EMT epigenetic repressions of CDHI, which encodes E-cardian is mediated by the recruitment of EMT inducing repression factors snail to the CDH1 promoters, leading to a repressive mark. Further to this, snail can associated with the MI2 nucleosomes remodeling and decapitulase repressive complex, which can repress activity via deacetylation of CDH1 promoter. Yeah, ma'am, next one. Replicative immortality, alternative telomere lengthening. Is the telomerase independent? Sorry, ma'am. Telomerase independent homologous recombination based pathway which cancer cells use to overcome the Hayflick limit to maintain the telomere length. An interplay between epigenetics and genetics mutation leads to perturbations of histone variants, and its specific chaperone protein is alpha thalassemia X-linked mental retrading protein and death domain associated protein, which impairs the incorporation of histone variants H3.3 at telomerase, discovering their heterochromatic state and facilitating ALT. Yes, ma'am. After epigenetics play a key role in angiogenesis, histone deacetylase have been shown to downregulate the expressions of non-hipal lindo and but promotes an increase in the hypoxia inducible factor 1 alpha and vascular endothelial growth factors, thereby stimulating angiogenesis by the suppressions of hypoxia responsive tumor suppressor genes. Yes, resting cell glioblastoma multiforme is a very aggressive cancer with a dismal prognosis. Promoting therapeutic surgery into induced tumor cells death via tumor necrosis factor related apoptosis, inducing lesion. Base therapy that binds to a human death receptor 4. How the epigenetic silencing via promoter methylations of DR4 attenuates the tumor. Where was it? Attenuates, yes, ma'am. Attenuates trial DR4 mediated apoptosis. Further evidence of epigenetics mediation resist to the cell death is resistance to anthracycline therapy in the acute myeloid leukemia due to impaired DNA damage response for the defective nucleosome remodeling as a result of mutation of epigenetic regulation DNA methyltransferase. Yes, ma'am. Next, yeah, immune evasion. Epigenetic is fundamental to normal functioning of immune cells. Antigen presentation through MHC class 1 is pivotal for CD8 plus 7 cell activity. The class 1 transactivator NLRC5 is a transcriptional regulator of the MHC class 1 genes by the promoter regions of NLRC5 is frequently methylated among cancer, resulting in the reduction of MHC class 1 gene expression. Deregulating cellular energetics. In order to adapt to a hostile micro their high metabolic needs, cancer cell glycolysis instead of oxidative phosphorylation to metabolize glucose even in aerobic condition. The central activators implicated in the glycolytic phenotypes are the PI3K by AKT or mTOR pathway, along which the MYC and HIF1 signaling tumor suppressors that repress this pathway, and namely PDN, VHL, and LKBI and prolyl hydroxylase are epigenetically silenced via promoter hypermethylation, contribution to deregulation of cellular energetics. Is there more? Yeah. Genomic instability and mutation. Faithful genome replication and maintenance of genome integrity are underpinned by epigenetic mechanism. Transposable elements are highly representative sequence of DNA in the human genome and have their own regulatory sequence allowing for the independent expression ability to alter the expression of neighboring genes. Since the activity has a high propensity to disrupt genomic integrity, these are usually silenced epigenetically, but this regulation is lost in cancer.
Okay, thank you.
Tumor promotion. It's just DNA demethylation triggered transcription of information related genes, including chemokine receptor 4 and serum amyloid A is in advanced clear cell renal cells carcinoma, contributing to tumor promoting cell cancer cells instructive information via epigenetic remodeling. And further, the cancer involved more than just tumor cells, so our bodies are more than simply an assemblage of human cells. This sobering through leads us to our third hallmark, the microbiome.
Okay. Thank you very much, Zain.
Thank you, ma'am.
It was about new hallmark, the new second hallmark, and about hallmark number 3 will explain for us the Naim Nimra. Naim Nimra, are you ready?
Yes, ma'am, I'm ready.
Do you have a presentation?
Yes, I'll share my screen.
Okay, thank you very much. Naim Nimra, please explain a little bit faster, okay? Shorter. Shorter, yes.
Okay, sure. Is it visible?
Yes, great.
Okay, so I'll be talking about the second hallmark of cancer, which is the altered microbiome. The recent scientific advances have significantly contributed to our understanding of the complex connection between the microbiome and the cancer. Our bodies, they are continuously exposed to microbial cells, both resident and transient, as well as their byproducts, including the toxic metabolites. Circulation of toxic metabolites may contribute to cancer onset or progression at locations distant from where a particular microbe resides. Moreover, microbes may migrate to other locations in the human body and become associated with the tumor development. Several case-control metagenomic studies suggest that dysbiosis in the commensal microbiota is also associated with inflammatory disorders and various cancer types throughout the body. Although the microbiome influences carcinogenesis through mechanisms independent of inflammation and immune system, the most recognizable link is between the microbiome and the cancer via the immune system, as the resident microbiota plays an essential role in activating, training, and modulating the host immune response. Immunologic dysregulation is likely to provide mechanistic explanation as to how our microbiome influences cancer development and cancer therapies. In this presentation, this is discussed that the recent developments in the understanding of the human gut microbiome's relationship with the cancer and the feasibility. The concept of the human body being a vessel for other microorganisms is well established. The microbial metagenome in our body outnumbers our genome by at least 100-fold. The microorganisms first appeared around 3.25 billion years ago, and over 1.25 billion years of coexistence with multicellular eukaryotes. The interaction with microbes has shaped evolution, as illustrated by the microbial control of host homeostasis. It has been estimated that nearly half of the metabolites in plasma are of microbiota origin, but the human microbiome plays a duplicitous role. Helicobacter pylori is nearly ubiquitous among humans, colonizing about 50% of the world population, having co-evolved with humans in an association spanning over 50,000 years. It may also confer protection against asthma, demyelinating diseases such as multiple sclerosis, tuberculosis, and inflammatory bowel disease. H. pylori has also been shown to modulate energy homeostasis via cooperation with gut microbiota, impacting on circulating metabolic gut hormones. In contrast with these beneficial roles, as a component of the gut microbiome, it is also linked to the 90% of the gastric cancers. The carcinogenicity of H. pylori is associated with the expression of vacuolating cytotoxin gene A, which is also written as BACA, and the cytotoxin-associated gene A, CAGA. The cytotoxin-associated gene A positive H. pylori promotes genetic instability via perturbation of the mitotic spindle checkpoint, causing the chromosomal instability and epigenetic instability. Increased level of DNA methyltransferases lead to the hypermethylation of MLH1, which is a key DNA mismatch repair gene, and have been suggested to mediate a mutated phenotype in a hit-and-run fashion, promoting tumorigenesis. It is worthwhile to postulate whether the Escherichia coli has a role in cancer, being among the first bacteria to colonize the gastrointestinal tract of neonates, and advocated to promote gut health in multiple off-the-shelf probiotics. Certain strains of E. coli harbor a gene cluster hybrid non-ribosomal peptide synthetase polyketide synthase island, which produces genotoxic colibactin. The colibactin has been described as a bacterial warhead, forming bulky unstable DNA adducts via alkylation. Among the epithelial cell lines, the PKS plus the E. coli, they have been shown to induce double-stranded DNA breaks and interstrand crosslinks. Then we'll be talking about the microbiome tug-of-war hypothesis. Well, this hypothesis of the microbiome species may underlie the long latency in the enterotoxigenic bacteriophytes fragilis, which promotes the colonization of the PKS plus E. coli, together with leading to genetic and epigenetic instability. Following this, the colonization by the pro-tumorigenic Fusobacterium nucleatum further promotes tumorigenesis by aiding in the development of an immunosuppressive microenvironment and seeding metastasis, whereas anti-tumorigenic bacteria act to prevent malignancy. The long latency period, which may ultimately lead to subsequent accumulation of genetic or epigenetic mutations and overt malignancy, depend on the balance between pro or the anti-tumorigenic microbes. The BFT further leads to activation of the beta-catenin signaling and induces the STAT3, which is the signal transducer and activator of transcription, activation, and the T helper 17 immune response. The ETBF, it modulates the colonic niche to select for the bacteria with a colonization advantage, inducing upregulation of antimicrobial peptide, lipocalin-2, that causes sequestration of bacterial siderophores. The siderophores are iron-binding complexes that are pivotal for bacteria to thrive in iron-limiting environments. Hence, bacteria which are resilient to lipocalin-2, such as E. coli, they begin to thrive along with the ETBF. Therefore, the first hit in our hypothesis of CRC tumorigenesis is orchestrated by the ETBF, which is followed by the co-colonization of the ETBF along with the PKS plus E. coli, which is followed by the Fusobacterium nucleatum comes into play. An anaerobic gram-negative bacterium, which is the Fusobacterium nucleatum, it is typically resident in the oropharynx, participating in the dental biofilm formation. Its virulence factor FADA adhesin binds to the extracellular domain of E-cadherin and promotes tumorigenesis via beta-catenin and WNT signaling. The F nucleatum is also immunosuppressive, causing inhibition of the T cells responses while allowing for the expansion of the tumor-promoting myeloid-derived immune cells. The FAP2 cells protein of the F nucleatum binds directly to the inhibitory receptor, which is the T cell immunoglobulin and ITIM domain, and it inhibits the natural killer cell activity, which leads to the immune invasion. In support of this hypothesis, the F nucleatum did not initiate tumor formation in vivo, but sustained the pro-tumorigenic momentum in the latter part of the multi-step CRC tumorigenesis and facilitated metastasis. Moreover, the treatment with the metronidazole, an antibiotic that reduces the F nucleatum load, it resulted in reduced tumor growth, suggesting that the tumor cells, they are rewarded for carrying F nucleatum by its modulation of the microenvironment at the distant metastatic site in the favor of tumor growth. Meanwhile, there is also a subset of anti-carcinogenic bacteria that include the Faecilobacterium roseburia and Slackia sp, which generate the catabolites such as short-chain fatty acids, for example, butyrate, and the antioxidant equol. The butyrate downregulates the pro-inflammatory gene expression and suppresses tumor growth via inhibition of histone deacetylase. The outcome of the tug of war, it depends on the epigenetic factors such as DIKE, which will give the final upper hand, aiding in colonization by either pro or anti-carcinogenic bacteria. The majority of the pancreatic ductal adenocarcinoma patients, they have a dismal prognosis, but a small subset of patients which survive longer than 5 years. These long-term survivors, they have a higher tumor microbial diversity with distinct tumor microbial signatures as compared to the short-term survivors. The tumor microbial diversity was shown to an expert and immune activating effect via improved immune cell infiltration to the tumor milieu. Furthermore, the colonization of the pancreatic tumors by gut microbia was identified with 25% of the PDAC microbial composition, matching that of the gut. Those patients, the data was collected from those patients who were long-term survivors into the tumor-bearing mice, led to immunoactivation in the murine tumor microenvironment and a significant reduction in the tumor growth, reiterating the role of tumor microbiome in disease progression and outcome, as well as the potential of the FMT in treating PDAC. The microbiome is more than bacteria. It is important, and we need to consider more than simply bacteria and viruses. The fungal infiltration from the gut into the pancreas was shown to occur via the sphincter of ODI, which serves as a direct link between the pancreatic duct and the gut. The taxonomic diversity analysis identified the dominance of the genus Malassezia in PDAC tissues compared to that of the gut in mouse models. Comparison of sequencing data of the patient fecal samples to that of paired tumor tissue corroborated these findings. The antifungal ablation with amphotericin B mitigated the pancreatic dysplasia in the mouse models and was shown to work synergistically with the gemcitabine in reducing tumor burden. Through population experiments, the Malassezia globosa was identified as being responsible for the PDAC disease progression, via the fungal-mediated activation of the mannose-binding lectin C3 cascade, and which is an immune system which serves as an opsone. Upon binding to the sugar motifs on the fungal wall, it triggers the complement cascade, in particular the C3, a pivotal component downstream of MBL. Based on the interference from the study, we can speculate that the diagnostic assay using the taxonomic composition of the stool samples may be appropriate for early detection of PDAC, and the antifungal therapy may be efficacious. More than 85% of the human papilloma viruses are cleared spontaneously, so why can't the remaining 15% mediate progression to cervical neoplasia? The answer is basically with the vaginal microbiome, the dysbiosis of which plays an important role in HPV-related cervical cancers. The lactobacillus species are dominant in the vaginal niche and are characteristic of vaginal health. They maintain the vaginal microenvironment in an acidic state, which is the pH is always less than 4.5, via the production of lactic acid and protect against invading pathogens such as herpes simplex virus, human immunodeficiency virus, Neisseria, gonorrhea, and E. coli. The depletion of lactobacillus species has been linked into an increased risk of exposition of the HPV infection, and its reduced clearance into lactobacillus dominance and increased vaginal microbiome diversity correlated strongly with the cervical neoplasia severity. The microbiome also plays an important role in deciding the outcome of both conventional chemotherapies and immunotherapeutic interventions. It can alter the bioavailability of the drugs and the DNA damage induced by platinum-based regimens, which is severely attenuated in the absence of commensal microbiota. The oral administration of bifidobacterium in mice controlled melanoma growth on a par with checkpoint blockade, used programmed cell death ligand, which is PDL1, specific antibody, and co-administration resulted in near eradication of tumor growth. The microbiome also has a role in immunosurveillance, as seen with the hygiene hypothesis, which link an increase in the incidence of some cancers to decrease in the exposure to certain microbes. In conclusion, the microbiome exerts both beneficial and nefarious effects over the human body. It also has a role in each of the triumvirate of the immunoediting, namely elimination, equilibrium, and escape during the tumorigenesis, and as such in a pivotal enabling hallmark of cancer. The antibiotic-mediated alteration of the gut microbiota has been shown to alter cerebral tumor microenvironment, thus affecting the glioma progression, which brings us our final enabling hallmark of cancer, the neuronal signaling. Thank you.
Thank you very much. The next hallmark will... What? Okay, next hallmark will explain the last one. The last one new hallmark will explain the rabia from groups G and 6. And I will show the presentation. Она попросила. Rabia, are you ready?
Yes, ma'am.
Захран. Что это такое вообще? Это что-то другое. Игорь Алексеевич, не могли бы вы...
Да?
Не могли бы вы, да? Спасибо большое. Быстрее. Быстрее выключаю. Давай. Избежать некоторых недоразумений. The new hallmark number 4, the name is like altered neuronal signaling. Rabia, are you ready?
Yes, ma'am.
Okay, start please.
But ma'am, I can't see the screen.
Да, пока экран не видно, то есть написано, что запустили. Записано, что запустили экран, но еще не видно экран.
Так. One more time. Все, да?
Да.
I can see. Now can I start, ma'am?
Yes, go girl.
Yeah. Hello everyone, good afternoon. I'm Rabia from GM6. So today my topic is about the hallmark 4 of the cancer. So I'm gonna start. Vicious, that is the writer, he write in his book that the corpus humanum fabulous libri spectrum describe the tender nature of the blood vessel and nerves. While inversions and blood supply being compatible for growth and survival, since agonesis has an established role in authentic to deliver further into the role of the nerves in cancer. A topic that is often overlooked, that is perhaps the reasons might be difficult involving in the observing nerves during routine histology of tumor specimen. But nerves are one of the most significant aspects of the tumor progressions. Metastasis involves in the central nervous system as well as the peripheral nervous system result in the manifold increase the mobility and molarity. Well, in 14 surgeons attempts to transcript the terminal nerves which runs along the face and accompanying blood vessels in order to cure the tumor of the lips, but they can't cure the patient. They failed in their exams, in their tests. So now, according to the recent advanced study, understanding of the functions of the nervous system, its roles in tumor initiated and progressions can be better educated to the deliver therapeutic benefits. But these density of the nerve fibers in tumor tissues correlate with the aggressiveness of the disease among the multiple cancer, including the breast cancer, lungs, correlate, and prostate cancer. Based on these observations, one might advocate neural transcript to control the tumor progressions. However, then when the PDAC mucous model subdiaphragmatic vagometry and targeting the vagus nerves, a mixed nerve with both sensory and peripheral system component, and resulting in the encoded tumors growth and reductions in the survival, contributing transcriptions of the some of the nerves in gastric cancer models, resulting in the suppressions of the tumorigenesis. Instead of the radical approaches of the transcriptions of the alternative approaches is used to chemical derivations as performed in the specific targeting of the sensory nerves in adult occurring and used of the specimens inhibiting pancreatic duct, that is PDAC and progressions. Another approaches is where injections is botulinum toxins of A or neurotoxins into the gastric wall. The inhibiting progressions to avert calcomania amongst the pancreatic models and inhibit the disease progressions in advanced gastric cancer models. Next one, ma'am. Beta blockers for inhibiting tumor progressions. Sympathetic nerves are implicated in the blood vessels and penetrating during the early development. Sympathetic nerves release neuroadrenaline, the circulating levels of the which increase the during chronic stress. Beta androgenic receptors mediate most of the effect of the neuroadrenaline. Chronic stress has been attributed as a risk factor of the cancer. Reproductions of the effect of the chronic stress is transgenics mucous model of the breast cancer where the long-term administrations and non-selective beta allergenics against resulted in administrations of the irritable lines and then the lymph increase the lymph node metastasis wells inhibit the adrogenics signaling with propanol or non-selective beta blockers result in inhibiting of the metastasis of the lymph nodes. Well, ma'am, next one. Ma'am, next slide. Okay, perineal visions in the pancreatic ductal adrenal conment. When the perineal visions in link to the worst progenesis in PDAC with PDAC cells resulting nerves with the nerve growth factors. Nerve growth factors in immune PDAC models, chronic stress development dependent sympathetic nerve signaling targets further tumors growth where the feed forward lobes where adrenogenic signaling stimulate NGF, which promotes the further inversions of the tumor cells, where the exogenesis resulting in the increased neuroadrenal accumulations in the tumors and microcore environments, including the B2 adrenogenic receptors, depending on the PDAC progressions. Next one, ma'am. Ma'am, next slide. putting the cart before the horses, and where the nerves migrate towards the tumor or tumor cells migrate towards the nerves. So there is a blockage of the beta-2 adrenergic receptors or NGF receptors, sympathetic interactions between the brain tumor cells and neurons. Closures the interactions. Schwann cells, that ganglion cell responsible for the myelinating peripheral nervous systems, are key the neural homeostasis, participating in the vagal degenerations, neural repair of the regenerations. In via the models using the red cystic nerves, Schwann cells display a high affinity towards the pancreatic and the clone tumor cells, but not normal cells. Migrated towards the tumor cells, thereby the outlink pathway of the tumor-derived neurogenesis, nerves growth factor, and its receptor TRKA and P75 and TR are article regulate of the gland inversions and neural outgrowth. They are also incompleted in the neural tracking the ability of the tumor cells to migrate along the axons. Ma'am, next slide.
Also next.
Yes, ma'am. Based on the both.
So I'm sorry. Based on the both of the studies.
Were the anti-epileptical drugs that is parasympathetic, such as the levetiracetam, might inhibit the glioma progressions. Alternative approaches could be non-competitive AMPR, MPAR antigens such as the progluent, which has good penetrations to the brain for use of the glandular treatment. Ma'am, next one.
This one?
Prostatic. Yes, ma'am. Prostatic therapeutic synapse. Triple negative breast cancer carried a poor prognostic at the late to the expression targetable hormones receptors and the human's epidermal growth factors receptor 2 captured by the propensity to the menstrualize to the brains, while the mental and de-aspirated receptors and MDAR attached of the glutamate receptors play a key role in the synaptic plasticity of the central nervous system, but has also been multi-related evidence.
Next.
Okay, ma'am, next one. Yes.
This one?
No, ma'am, next. Okay. Nerves and the tumor microenvironment. Well, nerves can also play a role. Nerves and neurosignaling during the tumorigenesis by the orchestins or an immune suppresses tumors microenvironment. Beta-2 adrenergic receptors signaling by adrenergic nerves can inhibit lymphocyte egress, effectively reduce the recruitment of the agent and permit the T cells. Then the manipulations of the autonomic nerves use the novel viral vector-based neuroenerging technique and revealed the accumulated breast cancer progressed with the sympathetic nerve stimulations in the tumor, while the local sympathetic variations curtailed tumor growth and reduced the expansions of the immune checkpoint molecules such as the programmed that one PD1 and PDL1, as well as the FOXP3 that mediate the immunosuppressions, such as the strategies of these localized invasions targeted neural input using genetic neuroimaging techniques may hold promise to stimulate the immune system while offsetting the deleterious and side effect of the systematic use of the checkpoint inhibitors.
Finish?
And ma'am, now last one.
Okay.
That we have the whole material. Conclusions. The understanding of the cancer finally, the hallmarks of altered neural signaling consist of there are two hallmarks proposed of the conference.
I'm sorry.
Ma'am, last one.
Yes,
Just
this is the last one. I'm sorry.
Finally, the hallmark of the altered neural signaling consists of the multiple cluster, that is metatastis. Then two factors which cancer cells use to design their microenvironment, their advantages, microbiomes, and the nerves. Tumor cells use nerve to establish blood vessels and garners proliferative cases. Cancer can be associated with the excruciating pain or keep being that cancer cells recruit numerous cells, a trait that can be impacted the pain management. Two modalities for managing the hallmarks of the alternative neuron signalings are either to include the resections of the nerves in surgical proctors of the tumor's management, and then to target the nerve's growth factors, localize the inversions of the neural signaling with the tumor microcoal environment. Future studies may look into the possibilities of the targeting arguments which has an established into the migrations of the sympathetic precursor, and consider cancer as a conductor of the malignant symptoms and the hallmark as a miscreants. And we need to tone our hearing to appropriate every key nuance of the piece. By identifying the new performers, we can adopt our inversions, re-editing or the occurs and the established the rhythm of the life. So that's it, ma'am.
Thank you very much, Rabia. And now we'll finally go to the chapter about the treatments and researches. And we will start with a little history about how treatments were discovered and evolved during the time. And the first guy who will be representing you that will be Amin Bakhrul. Amin Bakhrul, do you have this presentation by your side?
Yes, ma'am. Can
Okay.
I?
Can you open that? Towards before you start, try to make it really a little shorter than it is because it's quite a lot.
So, can you see my screen, ma'am?
No, blank.
Now.
It is blank. Try one more time.
Now. Ma'am, can I start?
It's not visible. Can you restore that again?
Okay, ma'am. So please share my presentation, ma'am.
Okay. One moment then.
Okay, ma'am. So today, the next one, ma'am. So today, my name is Bahrul Amin. I am from Group 7. Today, my topic is new approaches in treatment of different types of cancer and timeline in cancer research and discovery. So, ma'am, I think you can share the first one, my presentation.
Can we skip to the history? What?
Ma'am, I think you can, now I can send you a presentation. This my presentation, ma'am.
No, 1 second. I'm sorry, 1 second. I will.
Okay, the last one, ma'am.
Last one, okay.
Now I can send. Ma'am, now I can send in WhatsApp group.
No, we got it.
The third slide, ma'am.
This one?
Third, next.
the slide number.
Okay, ma'am. So there are some commonly used name in this slide. So neoplasm. So it is like a new growth or in tumor. It is a swelling caused by neoplasm. So and also begin neoplasm. So one which is restricted by its capsule does not separate to other areas of body. And the next one is malignant. So and which separate of different part of the body. So they are also included in next some commonly. Next, ma'am.
I'm sorry. I'm sorry.
Okay, ma'am.
Uh-huh.
So it is also the history. The oldest description of cancer are dated back to 1600 BC in Egypt. So professor described 8 cases of ulcer of breast treated by by tool. He called the pair drills in 1761, mentioning the first performing autopsies. He studied post-mortem pounding and suggested cause of diseases. This leads to the foundation of scientific study of cancer oncology. The next slide, ma'am. So eye of cancer surgery. So the famous Scottish surgeon Lord Hatter, he is the most popular scientist in 1728 to 1793 suggested that some cancer might be cured by surgery if the tumor had not invaded nearby tissue. It was movable. He said this is there is no impropriety in the remove it. So he conducted a lot of autopsies and studied cancer. The next slide, ma'am. And cancer research, the origin of the word cancer is created so to the Greek physician so Hippocrates so that lived from 416 to 370 BC, and he was considered the father of modern medicine. So cancer has indeed been around from quite some time. So with the earliest known description, so appearing as far back as 3000 to 1600 is severely improper. So from ancient description cancer as well as producer to remove breast tumor by cauterization. So but it was not until 16 to 17th century so that it become more acceptable for doctors so to discuss bodies and investigate these malignant lengths of cells in the body. From then on, our knowledge on matter has kept growing in trophies to fight the parasitic cells has been the light of the day. So the next slide. Then the first milestones in cancer research and discovery. So in this during the past 250 years, so we have witnessed many landmark discoveries in our effort to make progress against cancer and efficient known immunity for thousands of years. This timeline show a few key milestones in the history of cancer research. The first one is
Amin. 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 Okay, she's gone. He's gone. Now, shall we wait? Нам подождать?
Sorry, ma'am, can you hear me?
Oh, yes, now it's fine. Okay, continue.
In 1886, at the inheritance of cancer risk. In 1895, the first X-ray discovered, and also 1898 iridium discovered. In 1902, the cancer tumors in single cells with the chromosomes damage. The next one is the next slide, ma'am, please. Then they show in this diagram the research milestones in cancer research and discovery, and the next slide is also shown in this diagram. The next slide, ma'am, please. And then 1803, the first use of radiation therapy to cure cancer. In this picture, also showing the showing the first one is the doctor has radiation therapy. And the 1909 immune surveillance in 1911 cancer in chickens. So in 1915 cancer in rabbit. So in 1932 the modified radical institution for breast cancer. In 1937 the National Cancer Institute discovered. In 1937 breast spring surgery followed by radiation. So in 1941 humoral trophy. In 1947 anti-metabolites. So in 1949 nitrogen mustard. In 1950 cigarette smoking and lung cancer discovered in 1950. And he is the most common people disease, and that's why the most common cause of cancer. In 1953 the first complete care of human solid tumor. In 1958 combination chemotherapy, and this is also most important. The next slide, ma'am. In 1960, the Millosby chromosomes. In 1964, the focus on cigarette smoking. In 1964, the instance of Bar virus discovered. In 1976, the DNA of normal chicken cells discovered. In 1979, the TP53 genes discovered. In 1984, HER2 gene discovered. And also the 1984 HPV 16 and 18 discovered. In 1985, breast surgery discovered. In 1993, the FOVT discovered. In 1994, BRCA tumor surprise gene cloning discovered. The next slide, ma'am, please. In 1995, though, also discovered surprise gene cloning. In 1996, discovered it. So in 1998, NCL sponsored breast cancer prevention till discovered. And 2006, NCL study of STAR discovered. In 2009, Krivakis discovered. 2010, the first human cancer treatment vaccine discovered. In 2012, for the NCL screening till discovered. In 2014, analyzing DNA in cancer discovered. In 2014, Credis 9 discovered. In 2016, cancer moonshot discovered. And this also in this prevent in the trophic cancer vaccines in the show in this picture. And 2020, international fan cancer discovered. And the next slide, ma'am, please. Whole genome. Okay, ma'am. The next slide. And this diagram also showing the different pictures, ma'am. The next slide. Timeline and cancer research. So cancer rates continues to increase, particularly in the developed world. Many cancer are associated and targeted therapeutic. So one approach to avoid side effect to targeted only cancer cells and not healthy care. So in 1906, Blue Engine continues, and he is a magic bolt discovered, and he is the most popular to describe drug that specialized targeted diseases caused by organisms. So magic drug become a reality in 1970 with the development of monocloning. So antibiotics. So in which are designed so to ligand scientific found only on cancer. The first one is 1990 United States approved that to date 12 ABS have been approved to the treatment of cancer. The most recent being for the treatment of lymphomas. The next slide, ma'am. Sorry, ma'am. The next slide. Active immunotherapy. And this is also known as a cancer vaccine. So stimulates a patient immune system. So and so, ma'am, please previous slide. Patient's immune system to destroy cancer cells in 2010. Ma'am,
Yes, yes, what happened?
ma'am, I think but
Continue. I turned off.
mute kar do.
Что делать?
Проблемini sir khatam kar.
Bahru, which slide you want?
Akhirni round.
Ma'am, the next one.
Next one. Active immunotherapy. Sure.
Target, ma'am, the who is saying the song.
Is it fine?
You
I deleted it, so you can continue.
Ma'am, who has entered? Okay, ma'am. So in this 2010, the FDA approved the first such vaccine, and this is the first vaccine for cancer in 2010. So and treated advanced hormones, so different cancer, because vaccine targets an immune response. So they may protect the patient against the metastasis and relapse. So in which and with the other side effect profile is generally mild, primarily limited to inject site inflammation. And the next slide, ma'am, please. Ma'am, next slide, please.
Which one?
The next 18. Yes, yes,
18.
18. I put it already. Okay.
Okay, so this in this new of what is so there are different throatic cancer, and how do we treat organ cancer? So currently approved surgery and what happened? Personalized medicine, so and gene expression, so diagnosing and biomarker can be used to strictly patient into patients and non-responsible to select patient who are less likely to suffer side effect. So this approach will help physicians or discuss with marketing drug will be helpful specific patients. So clinical trials also can be exaggerated normally to the through cancer imaging, so therefore increase the cancer of early diagnosis, ma'am. So next slide. Next slide, ma'am. Next one.
Which one exactly about this?
Yes, ma'am. What is throat cancer? It is a refer the continuously tumor of develop into the
I mean,
yes, ma'am.
are you going to tell about the ovarian cancer or which one? Or throat?
Oh, this is okay, ma'am. Do not change the slide, ma'am. Now it's okay.
I'm sorry. My bad. My bad.
Repair the continuously tumor that develop into the throat-like pharynx. So and why so your throat is muscular tube, so that begin behind your nose and end in your neck. Type of throat cancer, it is a different type. So and the first one is the nasopharyngeal cancer, and this begin the nasopharyngeal, the part of your throat just behind your nose. And the next one is hypopharyngeal cancer, and this is the begin in the lower part of your throat, just above your esophagus, and glytic cancer, and begin into the vocal cord. And the last one is supraglytic cancer. This is begin in the upper portion of the larynx, and cancer that affect epiglottic, so the epiglottic that block food from going into the wet tube. The next slide, ma'am. Next one, ma'am. Ma'am, next one.
Symptoms? No.
Symptoms. So yes, ma'am. No, it's okay, ma'am. Septum. So it is a constant need to clear your throat. So and like a cup make up blood and swelling lymph nodes. So in the neck, envisioning the air fan or swelling up the eye, chew and throat or neck hardness. So treatment of throat cancer. So it is a different treatment like surgery. So in chemotherapy and radiation therapy. The next one, ma'am, please. Ma'am, next slide.
Ovarian cancer? No.
What is ovarian cancer? It is the most common type of ovarian cancer that start from the epithelial cell gland from the adenocardiomia. Other common adenocardiomia is found in the and uterus. So sometimes cervix. So other type of ovarian cancer start in the egg. Tumors. How do you? The changing in bowel and bladder function, so pelvic abnormality, pain, so decrease the epididymis symptoms, so and increase over time, so using more frequently and vaginal bleeding. The next slide, ma'am. The next slide, 22 number, ma'am. Please, next slide.
Мне кажется, нужно сказать, что I mean.
So yes, ma'am. The next slide, 22.
One moment, please. I've noticed that you describe really nice things. You describe different types of cancer, how we treat it, and all the conditions related to that. But can we, if you do not mind, can we skip this part because we're actually trying to focus on the therapy? Is it fine for you?
Okay, ma'am. Okay, ma'am. No problem.
Okay, so we are just roughly just go to the treatments. Is it fine?
Skip part.
Okay, so maybe I can go for the last slide and then you can nicely finish that.
34, ma'am.
34.
Causes of leukemia, ma'am. 34.
34, yes. Wait, please.
Causes of leukemia. Yeah, causes of leukemia. Experts say that the different leukemia have different virus been seen in some alkalizing used in the previous cancer, hair drying, and genetic. Some studies research famously history and looking at the twice have some people have higher risk of developing leukemia become of the single of multiple genes. So and the next one is people with Down syndrome, so have a significant higher risk of the leukemia compared to people who do not have Down syndrome. The next slide, treatment of cancer. And it is some of the common evidence of treatment of the other than the chemotherapy. The treatment of our blood cancer, including attempt the stem cells and bone marrow transplantation. So and herbal deals leukemia, blood cancer care. So and certain herbs are also used in the cancer. And the next slide, ma'am. Timeline and cancer research. Timeline and cancer. So there are today animal experimental and mice and other animals so continues to be at the forefront of cancer research, playing a key role in the development of new therapies or the development of technique to modify the gene of mice, and has led to a great deal of interest of them for cancer research. And recently study of GM mice has really important into the ferrous, particularly type of pancreatic precaution and increasing understanding of involved many cancers such as the role of modern day cancer research, and there are still much to be discovered and much to apply to beat and this kill 9 to 8 million people in 2018 alone, and affected a million people. All, ma'am. There are also. Thank you
very
for
much.
your attention, ma'am.
Thank you.
Thank you, ma'am.
Oh my God.
Okay. Okay.
Okay, thank you very much. I mean, good.
Thank
Next
you, ma'am.
one.
Now we will step into the therapies, which are new ones, comparable new ones. So we will start with Imadullah, who will introduce you to the immunotherapies and monoclonal antibodies and some examples of the drugs that we use.
Yes, ma'am. Can
Imadullah?
you share my PDF, ma'am? Presentation.
No, this is yes.
I'm sorry, one second. Good. Let's see. This one?
Yes, ma'am.
Okay, please start.
So in the treatment of the cancer, ma'am, there are different types of therapies used with one type is it is immunotherapy and the other type is chemotherapy, ma'am. So these two therapies are used in the treatment of the cancer. The difference between the chemotherapy and the immunotherapy is that they both use the drugs to inhibit the replication of the cancer cells, ma'am. But the difference between the chemotherapy and the immunotherapy is that the chemotherapy target the main replicating cell and also inhibit the neighboring cell of the cancer and also inhibit the healthy cell. And instead of the immunotherapy, ma'am, so immunotherapy doesn't inhibit the healthy cell, it only targets the replicating cell, ma'am. So immunotherapy, from the name, it is just as from the name, so it is the type of that treatment of the cancer, ma'am, and which enhance the immune system of the body and which help the body to fight against the cancer cells in the body, ma'am. And what is immune system? So immune system is the defense line of the human body, which is consist of different types of complexes, which is just like T cell and B cell, and help in the body to defend against itself the cancer, ma'am. So there are different types of the immunotherapy, ma'am, and it is also consist of just like monoclonal antibodies and tumor agnostic treatment, ma'am, such as nucleotical virus therapy, T-cell therapy, vaccine, and there are different types. We use the different types of these immunotherapy to different types of person, which is depend on the body of weight, their health, their size, their location, and how we use it in these type of immunotherapy plan. So how these monoclonal antibodies and tumor agnostic treatment works, ma'am. So the difference between the normal antibody and the monoclonal antibody is that normal polyclonal antibody is formed by different types of parental cells from lymphocytes, and in the monoclonal antibodies, in the case of the monoclonal antibodies, ma'am. So these are formed from just one type of parental, so therefore it is called monoclonal antibodies, ma'am. And as we can see there, there are two types of there are two types of chains which is found in the structure of the antibody. So the first one is the long chain, and the other one is the short chain. So the difference between the other antibodies and the monoclonal antibodies, that they have the epitopes, they have special types of antigen binding sites or antigen recepting sites, so they could attach to the cancer cell. Then can you change this? Yes, ma'am. So, ma'am, there are different types of checkpoints which is present in the body and the lymphatic system, which check the replicating or the invading cancer cell which is present in the body and moving through the body to different sites, to different parts of the body. Because as we say that the malignant tumor can be caused anywhere in the body, just like in the breast, and the throat and the lungs. So it is moving in the body. So there are different types of checkpoints which is present in the immune system, and which is normally act is that to stop the immune system to respond differently and prevent these to prevent these cancer cells and stop attacking the healthy cells of the body, ma'am. And these checkpoints checks the healthy cells and the mutated cells which is mutated by the cancer cell, and they stop these mutated cell, and then the T cells comes in attack on and the T cells and the other monoclonal antibodies attacks these types of cells and kill them, ma'am. So there are certain types of side effects, ma'am, which depend upon the person, because we know that the immunotherapy is just a traditional therapy, ma'am, and there are different side effects of these immunotherapies, ma'am, can be caused different types of. It depends on the person how he gets affected by that, but some of these are can be caused by weight loss, ma'am. It can also cause rashes on the skin, ma'am, sore in the mouth, ma'am. It can cause you that bleedy blistering on the skin, hair loss, ma'am, loss of the melanin and the non-pigmentation can be occur, ma'am. Itchiness can be occur in the skin, ma'am. And these are the specific therapies, ma'am. And the other one is the ma'am, can you change the slide? Because I'm the third slide, ma'am. So the non-specific, as we already discussed about the specific, and there is a subtype of non-specific immunotherapies, ma'am. So our system is our body contains a sub type of protein which can help the body to defend against itself when the antigen comes to the body, and these type of radiation therapies can be worked. So the first one which act against the cancer cell membrane, which is called interferon. So it is, from the name, it is mean that it interferes in the developing of the cancer cells. It is a special type of protein which produced by our own immune system, and which alert our body to that there is some type of foreign particle or replicating, or typically some antigen or foreign bodies enter in your body, and it can be it can damage your body. So also interferons can be made in artificially in laboratory, and it can help your immune system to grow and slow down the growth of the replicating cancer cells and affect the interferon symptoms, ma'am. And the other one is the interleukins, ma'am. So interleukins is also the same type of the protein, but interferon is acting on the target cell, and Interleukins are the interferon triggered cell, which triggers and helps to communicate with the interferon and response and help the immune system to respond to the cancer cell. So the other one is the ecolytic virus therapy. So how we discovered this virus therapy? So as we know that viruses is just like obligate crystal thing which cannot reproduce by itself, because when it's outs of the body, so it cannot replicate because it has not its own machinery of replication. So it is obligate to the cell because it needs a host to replicate itself again and again. So that doctor inducted that though genetically it can be the modified virus enter into the to the cell and make it so it takes its the DNA and cause mutation in the DNA, and then it's produce its own viruses and replicating itself from that mutated DNA. So when in 2015, ma'am, the FAD approved that the first nucleotic virus therapy that to treat the lateral stages of a melanoma, ma'am, that cannot be treated by the surgery. So how they treated this type of disease, as we know that they removed that causing gene which mutated in the host, which the virus inserted in the host cell, ma'am, as you can see in the picture, ma'am. The other one is the T cell therapy, ma'am. So T cell therapy, as we know that the other one is the T cell therapy, ma'am. So as we know that immune system consists of the two main types of cell. The one is the T and the other is B cell. So T is also a killer cell which can target the cancer cell and kill it, ma'am. So it's also with if it should be enhance the performance of the T cell, and the enhancement of these T cell can be the T cell can be very good for the human body because it will run through the body through the blood and protect the body from certain antigen receptor, ma'am, and the cell and these receptor will not the receptor will to recognize, ma'am, the cancer cell which is also be mutated and can be very good for the body, and it will find the destroy, and it will destroy, ma'am, when it found the cancer cell, it will destroy the cancer cell, ma'am. And this is also known, ma'am, is symmetric antigen receptor T cell therapy, ma'am, and it's also every just like every therapy it have also its side effects like ma'am, high fever, mental confusion, blood pressure problem, and seizure-ness, ma'am. And what is the role of the monoclonal antibodies? So we already described that the ability of these cancer cell, ma'am, to evade, to replicate itself in the immune system, and it's the very dangerous property is this: the malignant tumor metastasis, ma'am, it is responsible for most of them is moderately, ma'am, and mortally associated with the cancer, ma'am. So these metastasis is a multiple step process which, ma'am, include consist of the cell proliferation and release of proteolytic hormones, ma'am, motility and evasion, ma'am, angiogenesis, ma'am, and supportive myoculation at vary the site of the growth of the cancer, ma'am. And how it's the anti-cancer, ma'am. So how these checkpoints, if we discuss about the checkpoints at the case of the cancer, so ma'am, it's also reverse that the resistance to the body to the immune system, ma'am. It's however in the late 18th century, ma'am, William Coley and others clinical investigation may observe that there is a solid tumor register, or we are there totally eliminated in the patient that suffer from the streptococcal skin infection, or we are there injected with the bacterial extraction. But unfortunately, ma'am, he didn't have any adequate control to, or the investigator didn't have the adequate control to that period where they were unable to duplicate his observation, ma'am. And the anti-tumoral and monoclonal antibodies, ma'am, so as it's such engineered and humanized type of its mechanism action against the cancer, ma'am, and it's also the mechanism is recently by the approved by the FAD, ma'am, that to target and kill the cancer cell, ma'am, and these novel also, which is these novels also these are also consisted of the novels, ma'am, the novels and which is target the CD52 and upon the were expressed on the malignant lymphocyte, ma'am, and granulocyte and macrophages and natural killer cells, ma'am. In addition to that, basically the treatment of the various cancer, ma'am, the mechanism of these the monoclonal antibodies, ma'am, used to deliver the radioisotopes, ma'am, selected to the cancer. Example that these which is the murine antibodies fragment in labeled with the metastatic cancer, metastatic colorectal cancer to extend, ma'am, the metastatic development. And what is the future of these the monoclinical mechanism of these the action of the mechanism of these action of monoclinical antibodies, ma'am? So one of the major problem is used to these is the cancer is constantly mutating them. So it changes, it also causes new mutant to itself. So it is very difficult for the checkpoints and also for the monoclinical antibodies to know the mutant cells. So it is very difficult for these types of checkpoints and monoclinical antibodies to recognize the mutated cell, ma'am. And also some honorable mentions. So the doctors who won the Nobel Prize in the recognition discovery of these immunotherapies with the T cell therapy. So there are two types of scientists who won the Nobel Prize award in 2018, ma'am, and the Nobel Prize ceremony in the physiology or medicine, ma'am. The first one is the James Parker Ellison, PhD of MD and from the Anderson Cancer Center, ma'am, in Texas, from America. And the second was the Tashuku Honjo, MD, PhD of Kyoto University in Japan, and honorable early today at the Nobel Assembly in the 1990s, ma'am. And the drugs which is used for the treatment of the cancer, so some of the drugs which is used for the cancer also used in the chemotherapy and the immunotherapy. So some of these drugs which is keep the copies, keep the cancer cells to not replicate or not to make the copies of themselves, and they were called the alkylating agents, ma'am. And the oldest types of these is the just like leukemia, lymphoma, Hodgkin diseases, man, and myeloma, and well is breast, lung, and ovarian cancer, which also Barulamin discussed, ma'am. So these are the cancer and the drugs which is mostly used for these types of used for these types of cancer, and doctors often used to treat the leukemia cancer and the breast cancer and ovaries cancer with the drugs which in the group include 5 chlorouracil and 6 meracatopurine and cytarabine and the methotrexate among many, ma'am. And steroid that can also act just like can stop these type of invasion in the body. So some of the steroid that can only our body can be produced, and it also can be produced in the artificial way, ma'am. And they can keep them having nausea, ma'am, vomiting after the round of chemo, ma'am, and also prevent, ma'am, allergies reaction to some of the drugs. Some of the steroid which doctor might precursor, ma'am, just like the pedinosine, ma'am, and the methylene prednisolone and the dexamethasone, ma'am. Thank you, ma'am.
Thank you so much, Imadullah. That was very useful and interesting presentation, really. So now, alongside of the immunotherapy and monoclonal antibodies and surgical and other types of the chemotherapies, we also have got therapies based on the nanostructures, nanoparticles, which will be presented by the Mumtaz Tahrim.
Yes, teacher. Allow me to share my screen.
Okay. Let's. 1 second. Да. 1 second. Ah, you will. Okay, TikTok. Ah, this is not you? Okay. Что
Teacher
делать?
will
Okay.
see my mistake.
So
Okay.
it was your TikTok, yes? Okay. I'm sorry.
My mistake. Okay, so should I start? Is
Yes,
it
please.
clear, the screen?
Not exactly.
Okay.
Yes, fine.
Fine now? Okay. My topic is a nanoscale and nanostructure based therapeutics. Chemotherapy, radiation therapy, and surgery are the most common types of cancer treatments available today. More recent treatments include biological therapies and vaccine, gene therapies, etc. But today we'll focus on targeted cancer, hyperthermia, photodynamic, and gene therapies. These therapies can be used in isolation or in combination with other cancer treatments, thereby taking advantage of the ability to target tumors actively or passively to respond to physical or chemical stimulation, internal or external, and to deliver therapeutic genes to the cell nuclei. The main objective of nanomaterials in cancer treatment is to deliver a therapeutic moiety to tumor cells in a controlled manner while minimizing side effects and preventing drug resistance. Compared to conventional therapies, nanoparticles show 6 clear advantages in cancer treatment or diagnosis. They can be synthesized in specific sizes. They can be engineered to target tumor cells. They can be engineered to penetrate cells. They can increase the plasma half-life. They can protect a therapeutic payload. They can be synthesized as a multifunctional platform for combined imaging and therapeutic applications. These are some examples of nanostructures materials with potential applications in oncology. The examples are shown graphically. Okay, then we have the nanoscale or nanostructured based therapeutic diagnostic agents currently marketed for the treatment of cancer. The drugs which improve drug solubility and bioavailability. These are some formulations, for example, deposit on etc. And the other polymeric formulations include genoxyl, on Casper, xenostatin. And magnetic hyperthermia includes nanotherm nanoparticles, and the photodynamic therapy includes oroshell nanoparticles. Okay, so now we have the controlled drug release. The brachytherapy product currently in development for the treatment of solid tumors, or the gladial treatment of newly diagnosed high-grade malignant glioma as an adjunct to the surgery. In vivo diagnosis, contrasts agent for MRI, Rezovist, which is nanoparticle coated with carboxydextran, are used. Okay, so now moving on to the main therapy, which is targeted cancer therapy. Nanoparticles are engineered to achieve cell targeting by using selective moieties which bind to its corresponding antigen, cell surface, carbohydrate, or overexpressed receptor in tumor cells. The rapid cellular proliferation of these cells is also exploited by coupling the nanoparticles with different biological agents, such as folic acid. The rationale for coupling these carriers with folic acid is the folate receptor overexpressed in a broad range of tumor cell types. Once it has reached the target, the cargo is released into the interior of the cell, and ideally, a signaling marker attached to the vector will aid the physician in visualizing the tumor. In addition to recognition moieties, carried drugs are signaling elements attached to nanoparticles. Numerous authors have also envisioned and designed vectors with additional functionalities. When using batch creators to synthesize nanoparticles, several drawbacks usually appear, including heterogeneous distribution of reactants and temperatures in the reactor, insufficient mixing, variations in the physiochemical characteristics of products from different batches, their inherent discontinuity, and the numerous post-synthesis purification steps that are usually required. Other continuous synthesis processes are usually preferred when synthesizing nanoparticles on a large scale. Okay, another concern is the adaptive response of the immune system after repetitive applications of the nanoparticles. Immunological memory created from the primary response to a specific nanoparticle provides an enhanced response to secondary encounters with the exact same type of nanoparticle. Finally, one of the last major barriers to achieve the transition of targeted nanoparticles is to use clinical practice. Next, we have the photodynamic therapy. Photodynamic therapy is also known as PDT. It is a technology that uses a photosensitizer. It is a technology which uses a photosensitizer that is activated upon exposure to visible or infrared light, and it transfers the energy to molecular oxygen, therefore generating reactive oxygen species. The photodynamic therapy is usually performed as an outpatient procedure and may be repeated and used in the combination with other therapies. The screen I'm sharing right now, the photo is like a small example of how the photosynthesizer and the tumor is used. The next therapy we have is hyperthermia. Hyperthermia is an anti-cancer therapy consisting of heating a tumor to inhibit proliferation of cancer cells with the aim of destroying or rendering them more sensitive to the effects of conventional protocols of radiation and chemotherapy. Hyperthermians usually use microwaves, ultrasounds, and radio frequency, which can be focused and used locally. We have now photodynamic therapy. The light source and the bacteria and the nanoparticles for hyperthermia settings show the advantage of being able to achieve high specific tumor targeting through the aid of an external magnet. These nanoparticles are selectively heated by a frequency alternating magnetic field. Okay, the next we have is gene therapy. Gene therapy to treat diseases DNA, RNA, or small interfering and antisense nucleotides into specific target cells using efficient vectors that aim to sustain stable regulated gene expression without creating unwanted side effects. The most widely used viral vectors for the gene transfer include AD, which are the adenoviruses, and the first clinical trials were approved in 1989. And during the 90s, numerous vectors carrying various therapeutic genes were engineered. Due to the complex nature of cancer, the numerous gene therapy approaches for fighting it mutant suppressor gene functions. the novel gene therapy trial conducted in 1991 on patients with advanced melanoma who received intrathecal injection of DNA. The results demonstrated for the first safety and feasibility of cancer treatment by gene therapy protocols using non-biological carriers. Cationic polymers have demonstrated superior gene transfer properties to those polymers having anionic or neutral charge at geological edge. 2 decades of cancer gene therapy using biological vectors, preclinical studies yielded satisfactory results in mild or long-term toxicity. Nanotechnology has already engineered powerful non-biological carriers of the variety of therapeutic genes that have demonstrated their efficiency. Since current knowledge of cancer cell biology is far from complete, ongoing and future clinical trials with these synthetic systems are expected to suffer and similar drawbacks from the experienced people. Also, the success of cancer gene therapies will be preceded by many failures, which will likely be due to a greater extent to our technological limitations and the flaws in the general concept.
Thank you very much, Tahim. Thank you very much. It was very interesting. And the last one.
Мухаммад Ильяс, здесь?
I think
Мухаммад Ильяс не отвечает на звонки.
Он сейчас на практике, наверное, из-за этого. Что задали делать ему. Ну ладно.
Ну все тогда.
Да, конечно, да. Закончим тогда нашу конференцию. So let's finish our conference. So technical problems come here. Some trolling happened also. So, but I tried to handle it, but still it leaks out. Okay, thank you for providing the conference. The record of this conference will be available in a website, I guess, now or uploaded to YouTube, so you can observe it again. So a few words to you, organizers, Aidana, ma'am.
Thank you very much for all students. You are prepared very interesting and important information, and thank you very much. You are amazing students. I'm very glad that I was here and listen your information. So again, thank you very much.
And special things I want to tell you for those guys who just come at the end, almost 1 hour left before the conference. You just took your part, and then you were such a brave, and thank you for that. It was really hard, and I just want to emphasize that you are really brave, good guys. Thank you so much.
And thank you for Rabia. She was very brave and agree for participating today. When she start preparing maybe 11 a.m. Thank you very much, Rabia. Thank you very much.
Okay, I also want to say my appreciation for all you. So I hope next the conference will be offline. So without this technical issues, without any kind of trolls, which we can control. Yeah. So also I want to mark Imadullah, yeah, your presentation so was very interesting. So you made it short in a short way. So but without reading, but explanation. So that's I do appreciate it the most. Okay, thank you.
Thank you, sir.
So actually every student tries their best. Okay, thank you. So for participant, we'll give you any certificate a little later. So get ready. Goodbye.
Okay.
Bye.
Thank you, sir. Bye, sir.
Have a good day.
So maybe
Have a
you
good day,
want to
sir.
express some thoughts about this conference, any suggestions? the students? No suggestions. Okay, then thank you. Goodbye.
Bye.
So next time, no PDF, sir.
Yeah.
Only verbal talking.
Yeah, agree with you. Okay.
Thank you, sir.
Good idea.